Hello, hi, good evening. Good afternoon. So this is Krishnaveni and we have been talking about the story of life on earth, and that's biology. So thank you so much for joining my session today. So on Monday, Wednesday and Friday, I teach for class 12, and for the remaining 3 days, that is Tuesday, Thursday and Saturday, I teach for class 11. So thank you so much for joining me today. So today it's Friday, so we are on class 12 topic. So we are almost done with 3 lectures of this topic, that is strategies for enhanced food production. So we've been talking about a lot of interesting stuff, how you can enhance the production in case of plants and in case of animals. So we are done with animals, so today we are talking about plants. So it was an interesting chapter where we discussed a lot of things. So Adil Rashid's future doctor is here. Ma'am, mentee morphology of flowering plants ka kab hoga? Okay, so we are going with animal kingdom. So let's finish animal kingdom and then we'll talk about morphology. Okay, so you want mentee on morphology of flowering plants. All right, so I will consider your suggestion. Yes, good afternoon, Priyanka. So it's not yet 4, so we say good afternoon. Okay, you want me to have mentee at 3 o'clock. All right, so 3 o'clock is my class. So you want me to have a mentee during my class. Then when will I have my class? Because I have to finish my portions here also. So when will I have my class? Alright, but it's considered. Okay, I'll consider your time. So you feel 3 p.m. is good time for you to take a mentee. Alright, and you want a mentee in morphology of flowering plants. Okay, considered. So not an issue. So anyways, thank you so much for joining my session today. So guys, please do like, share and subscribe. So let your friends know about the session. I guess again people forgot the lecture is at 3 p.m. today, because usually it's at 4 p.m. and for the last 2 days we are having it at 3 p.m. and yesterday, yes, people understood it's at 3 p.m. So today I guess people again forgot it's at 3 p.m. Yes, so please quickly remind your friends to come to the lecture. Yes. Yes, Priyanka says, yes ma'am, good afternoon. Yes, good afternoon, and thank you so much, Priyanka. Okay, so we'll talk more about your, yes, Keshav is here. So Keshav, why are you sad? Yes, hello, hi. So why are you sad? What happened? You're sad because the lecture is at 3 p.m. and you can't sleep any further? Is that so? Is that so you're sad? Hello, 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 yes. Alright, okay, so Keshav deleted the message. Alright, fine. Anyway, I saw the message and you had a sad smiley, so you're not interested in this topic, is that so? So don't worry, so once this chapter is over, we'll start your question discussion session for your class 12 as well. So we'll start off with the genetics, so that once again I will give you important ways to remember the topic as well as the questions, like how we started for class 11 yesterday. So we'll start for class 12 very soon, and we'll also have a lot of mentees. So henceforth you might have a mentee in Monday, Tuesday, Wednesday and not on all the days. Alright, so we'll have it on the first 3 days of a week. Fine. Okay, so let's go forward. So we were talking about the strategies for enhanced food production. So today will be the last lecture. We'll try to finish off today. So we still have to talk about your plant breeding techniques. So some parts are remaining. Then we'll have to talk about a single cell protein and then your tissue culture. So that's all we have to talk about. So we'll quickly talk about it. So what are the studies we were discussing? So first we started with your animal husbandry. Yes, then your plant breeding. Then we went ahead with your single cell proteins and then your tissue culture. So animal breeding, we spoke about how we can enhance the productions, how we can increase the productions of your animals, that is your livestock especially, right? So that was about animal husbandry. So under animal husbandry, we spoke on a lot of different subtopics if you remember. So what are those subtopics? Yes, the management of farm animals. We especially spoke about your dairy animals and your poultry. So poultry is a group of domesticated birds on whom you depend on meat or eggs. So guys, please don't eat meat or egg right now because the situation is very worse. You don't know what kind of virus is affecting them and you don't know whether the meat that you're eating is safe or not. So please don't consider. Okay, hemorrhoids, so you are very sad and fearful, so don't worry. So the more you get sad and the more you get fearful, you only tend to become more trapped to the disease, you tend to become more susceptible to the disease. Only when you say to yourself, everything is fine, I'll be okay, things will be fine. So the more you repeat saying, yes, yes, I am sad, I am fearful, then yes, your brain will also rely on, yes, we are actually sad, we are not able to challenge the situation. So that is the problem. So the more you say you're sad, you're fearful, that is what will actually happen. So try saying you're fine, you will be fine, and obviously you'll be fine. Okay, so this is one way of positive thinking. So repeatedly say you are fine, you will be fine, so definitely you will be fine. Alright, hello Vishal, hello. Yes, good afternoon, it's okay, you're late, so we haven't started the topic today, so it's okay. Alright, so we were just into the chapter and you are here. Fine. So don't worry, it's okay. Even there are people who have survived very severe fatal diseases. So this is nothing compared to that. You can easily overcome this, but have a lot of faith and determination. You're so young. So why are you scared? Why are you so fearful? You should be very brave. You should be in a position to explore a lot of things. It's okay. This is one of your experience. But don't be sad. The moment you're sad, you're getting susceptible to that particular circumstance. So don't ever be sad. So the moment you don't look sad, the moment you don't fear anything, then yes, you can definitely overcome the obstacle. The same with the case of your need. Yes, Harini, good afternoon. Though you're 6 minutes late, so still thank you for joining my session today. Alright, so Keshavan, take care and don't lose hope and faith. Alright, so then we went on to talk about animal breeding. So in animal breeding, we spoke about two different things. So what were the two different things? Inbreeding and outbreeding, right? So inbreeding gives you pure line cultures, and outbreeding, when you cannot have a lot of inbreeding due to inbreeding depression, we look for other opportunities, that is your outbreeding. So under outbreeding you have three things. First is outcross, then it is your crossbreeds, then it is your interspecific hybridization. Right, so these were the things that we were talking about. So you should remember each one. For example, inbreeding, you should know inbreeding gives you pure line culture. And what is inbreeding? When you mate a closely related species whose ancestors are common, you mate them, so that gives you an inbreed. So all the characteristics are the same, right? So that is called as inbreeding. At the same time, outbreeding, when you have differences between the two species that you are breeding, so it comes under outbreeding. So first type of outbreeding is your outcross. When you mate individuals who do not have a common ancestors for 4 to 6 generations, then it is called as outcross. All right. So to overcome inbreeding depression, we go ahead with your outcross. Then what is crossbreeds? When you have two different species who belong to the same class, you are going to mate them. So that is called as crossbreeds. For example, you have two different varieties of sheep. If you remember, we are talking about them. So they come under your crossbreeds. And the example that we were discussing is, if you take a Labrador and if you take a German Shepherd and if you mate them, that is called as a crossbreed. Then talking about interspecific hybridization. So what is interspecific hybridization? When you take two different species, when you take a donkey and a horse and you mate, it is called as interspecific hybridization. Alright, fine. Then what is beekeeping? So the maintenance of, that is also known as apiculture. The maintenance and rearing of bees is called as beehives or beekeeping, apiculture. So what do? You help, commercialize the honey that you obtain from here. Also it is used in the production of your bee wax. Right, then fisheries, so it talks about your fishing industry where you catch, sell and rear the fishes, so that comes under your fisheries. So under fisheries we are talking about a lot of examples of your freshwater fishes and marine fishes. If you remember, I showed you a lot of pictures of each of them. So with that we are done with your animal husbandry. Alright, so we were here talking about your plant breeding, we were done with animals. So talking about animals, yes, so when you talk about food, there are two sources of food. One is plants, one is animals. First we spoke about animals as to how to enhance the food production with your meat and egg. Now we are talking about your plants, because plants are a major source of food that we are dependent on. So how can we increase quality wise and quantity wise, how can we increase the production of your plant products? So that is what we are seeing. So that is plant breeding. Alright, so traditional farming, so this we already seen. So traditional farming can give you only a limited food supply, it cannot feed all the mouths. So hence we have to look for alternative techniques. Yes, so plant breeding is one technique that has increased the yield to a much larger extent. So one term that we have all heard about is green revolution. Yes, so green revolution talks about tripling the food supply, right? So dependent on plant breeding techniques, so we have developed high yielding disease resistant varieties in some of the basic crops, because wheat, rice and maize, they are a staple food. So in these type of food we have increased the yield, so that is a big success in this field. Alright, fine. So what is plant breeding? So plant breeding is a technique that you have used to enhance the food production. Alright, but how do you define it? So it is a purposeful manipulation of plant species that is done to create desired plant species. For example, if I need a plant of this particular characters, I am going to design it. Just like you go to a tailor and say, I need a dress like this. The dress should have so and so colors. The dress should have a knot here, a button here, a flower here. You give all those criteria, right? So similarly, when you say I need a plant which is giving a high yield, which is resistant to diseases, so we are going to design a plant like that. So we are going to play with the genes. So that is called as plant breeding. So how do you decide plant breeding? On what basis? You will design a plant based on whether it will suit the cultivation, whether it will give you better yields, or whether it is disease resistant. So classical plant breeding is done using crossing or hybridization of pure lines and then followed by artificial selection. So you cross two breeds and then you select the progeny which is having your desired characteristics. Alright, and then that is done to increase yield, nutrition and disease resistance. Alright. Okay, so when you're doing plant breeding, so when you're starting with the experiment of plant breeding, so what are the points that you should look for? So what are the criteria that you should look for? So the first thing, increased yield. So the first thing that you will look for is increased yield, whether your plant is giving you increased yield. What is natural and artificial selection? Okay, so natural selection is when your nature, when your environment selects a particular thing, that is natural selection. For example, during your pandemic, so survival of the fittest served, right? So one who were able to fight against your corona survive, so that is natural selection. The process is natural. Artificial selection is if I am pollinating two plants, if I am breeding two different species of a plant, it's going to give me n number of offsprings, each with a different characteristic. So a plant will have all the characteristics, a plant A and a plant B will have so many different characteristics, but I just want one of the characteristics from each plant to give me a better progeny, right? But it will give me a lot of it, but I'll have to choose the progeny which is having the characteristics that I desire for. So that is called as artificial selection, that is done manually. Natural selection is the one that is done naturally. Alright, it is a technique, it is a conceptual meaning. Alright, is that clear? Okay. So increased quality, then you look for improved quality as well. So increased tolerance to environmental stresses, salinity, so the increased salt condition, extreme temperatures as well as drought. Then resistance to pathogens, whether they can whether they are susceptible or whether they are resistant to virus, fungi and bacteria, because these three cause a wide range of diseases in plants. Alright, then increased tolerance to insect pests. So they are also one of the irritating problems that we have in agriculture, so because of which we had your Bt cotton developed, right? Okay, so now going further, so what are the steps that you will consider for a plant's breeding? So there are few steps that you will consider. For example, if you are going to prepare for NEET, again you look into the syllabus, you look into the weightage, you look into which book you have to study for, etc. So similarly, when you are going to do something with the plants, when you are going to play at the genetic level, you will also follow a particular type of steps. So what are the steps that you are going to follow here? Collection of variability, that is called as germplasm collection. For example, if I are talking, so we'll take the example of your staple food, we'll take rice. So there are so many varieties of rice, right? The rice that you use for food, okay, the normal rice, the white rice that you make is of a different type. The rice that you use to make idli dosa batter is different. So there are so many different varieties of rice. For example, you have basmati also. So you have so many types of rice. So first you will understand how many varieties are there. You will look for what is the genetic variability that is available within the particular variety that you're looking for. If you're talking about rice, how many varieties are there? You will look into that. So genetic variability you should study and you should understand. Once you understand the genetic variability, then you look forward to preserving the genetic variability. You'll try to preserve the genes of that particular variability. So some might be of the wild type that is present in nature. Some might be the cultivated type. Wild type is different, the one that occurs naturally. The one that you're cultivating in your farmland is different. So you'll look into that. So you'll collect and preserve the variabilities of a particular plant that you're talking about. So this collection of all the alleles of a gene of a given crop is called as germplasm collection. So you know what are alleles, right? Alleles are alternating pairs of a gene. For example, in case of Mendel's, it was capital T, small t. So that is alternating characters for your height. Yes, this is tall and this is dwarf. So when you collect this diverse collection of all the alleles in a gene is called as a germplasm collection. Ma'am, why virus-free plants are prepared from meristem cultures? Yes, we'll talk about it. So today we're going to talk about your meristem culture. Today we're going to talk about your tissue culture. So then we'll talk about why your meristems are virus-free. Okay, so guys, please do like, share and subscribe the channel. I know you have subscribed, please do share the link. Many people forgot the classes at 3 p.m. Alright, fine. So that is the first step. Now you have you have understood what are the variabilities that are available with respect to that plant. Now you will select a parent. So you have so many varieties in rice. Now you will select a variety that is giving you high yield. Then you will select a variety that is very, very tolerant to a lot of diseases. So you will select both of the varieties. Now you are going to actually evaluate whether they are strong, whether they are resistant to diseases. So that is how you check. Okay. Then you evaluate. So that is called as evaluation. Then you select the parents. Okay. So this is a superior parent. This is a superior parent. So we are going to breed these two. So that is how you select your parents. Alright, the next step is cross hybridization among your parent. For example, from this germplasm collection, you have selected plant A and D. So you want to breed these two. Now you will breed these two, you will pollinate these two, A and D. So it's going to give you n number of offsprings. From this, you would have chosen A because it is resistant to diseases. You would have chosen D because it's giving you a high yield. Okay, you just want two of those genes to come together and give you a progeny. But you will have n number of progeny. So from this, you will have to choose a progeny that is having these two characters. So that is called as cross hybridization among selected parents. And the next step is selection of the progeny that you desire. Okay, so you'll have n number of kids here. Okay, you will have to choose the kid with the character that you are desiring for. Fine. So that is selection and testing of a superior quality plant. Yes, Priyanka Singh, good evening and thank you so much for joining my session. Now, you have selected the progeny that has your characters of your choice. So, once that is done, then what you will do, you will actually test if it's supporting all other environmental conditions. If it is able to tolerate pest, if it's able to tolerate the environmental stresses, you will check everything, right? Then you will release it in the market, you will grow it on a large scale. So, that is how it is done. Fine. So these are the steps that you follow in case of plant breeding. So one example of plant breeding is your rice and wheat, your sugarcane and your millet. So we were talking about these in the last class. So your wheat and rice have your 𝗵𝗵𝗵𝗀𝗵 varieties. Okay, so wheat and rice. So the wheat production has increased from 11 million to 75 million. And this rice is from 35 to 89.5 billion. Alright, but how did they do during green revolution? They introduced semi-dwarf varieties of your rice plant. Okay, so what they did, they took, so these were the high yielding and disease resistant varieties of wheat, that is Sonalika and Kalyan Sona. You don't have to exactly pay attention to these terms, but just make a point, okay. So somewhere in an option, if they give you Sonalika and Kalyan Rayan, you should know they belong to your resistant wheat varieties, because if you want to eliminate the options, then also you should know what the option means, right, in that case. So this is the semi-dwarf varieties of your rice. Okay, so one of the high yielding variety was taken from your Philippines, that is IR8. Okay, so IR is your International Research of Agriculture. So from this strain was taken, and then you had another strain that was taken from Taiwan, and they were mated to give you resistant varieties. Alright, so then we spoke about your sugarcane and millets. So what about your sugarcane and millets? So Saccharum barberry is found in North India, but it had a very poor sugar content and it had a very less yield. The one that grew in South India, that is the tropical canes called a Saccharum officinarum, it had a very thick stem and it had a very high sugar content. Alright, so thicker stems and higher sugar content, but they were not able to survive in the North Indian conditions. Okay. Is IR8 produced in India? Yes, it is produced, but the original strain came from Philippines. Alright, Keshav? Okay. So they wanted to somehow modify Saccharum officinarum so that it can be produced in the North India. So they did a hybridization technique. So they cloned the gene that is giving high yield and a thicker stem, and hence made sure your Saccharum officinarum was produced in your North India. Alright. Then talking about your millet. So millet, we had your hybridized maize, jowar and bajra. So all these was developed in India. So what is the most beneficial part of your hybridized millet is that they were water stress resistant ones. Alright. Ma'am, when is the next test? Preeti Dubey. Alright, so we don't have a test today or tomorrow. So might be, I don't know, I will let you know. It's not there for today. Tomorrow I will let you know if there is a quiz. That is Menti. She's asking about Menti. Alright, so today there is no Menti. But please do subscribe my channel. If there is a Menti, you will get a notification. Alright, I will let you know. if at all we have a mentee, so we'll have it on a class 12 chapter. So I'll let you know on which chapter you're going to have a mentee. Alright, yes? Fine. So this is your sugarcane that we are talking about. So this is your Saccharum barberry. So this grew in your North India. And this is Saccharum officinarum. So this grew in your South India. So this has thicker stem and a higher sugar yield, whereas it was very poor sugar content and it had a thin stem. Alright. So they tried with the help of plant breeding. They were able to go Saccharum officinarum in the Northern Indian as well. Okay. Okay. Now, there was an objective behind your plant breeding process. Okay, so before you start something, there should be an objective behind it, right? So why are you doing, for what are you doing it for, what should be the end result, etc. So similarly, why was plant breeding actually started? To enhance, improve the quality, all that is understood, but practically where did you apply it? So that is more important. Is both used for sugar preparation? No, actually we use only Saccharum officinarum for sugar preparation. Okay. Yes, so we made Saccharum officinarum tolerable to the conditions in North India so that it is grown. Yes, yes. Now your second one is also grown in North India. Alright? Yes. So the objective behind your plant breeding technique, why it came into existence, is because it can give us disease resistant plants. So that is our primary objective of bringing plant breeding technique into reality. So you know, a lot of pathogens, they are infecting your plants. Your plants are susceptible to a lot of pathogens like your fungi, bacteria and a virus. So they affect the yield of your crop and they also affect the plant health as well, right? So this is very, very common, especially in your tropical climates where the climate is very, very variable, where it's very, very susceptible to a lot of changes. Okay. So crop losses are very, very significant, 20 to 30%, or sometimes the total itself is a loss. So in that cases, your breeding and development of cultivars resistant to disease enhances food production. So you know now this is the problem. So you have to bring out a solution for this problem. You cannot say, hurry, transport it from there, take it from here. It's not possible. Practically speaking, if you want to enhance the production, you have to look in for a solution. So if this is the problem, you have so many pathogens affecting it, and that is the problem, your yield is less, then you have to look for alternatives. So with the birth of genetic engineering and other techniques today, it is possible that you make the plant resistant to diseases. It's able to clone that particular type of gene into that plant, right? So hence we took forward plant breeding to make up disease resistant plants. Okay, it is native to Brazil, that is too tropical. Brazil is in the first position in sugarcane production. So you're talking about IR8. Yeah, but the original thing was obtained from you're talking about sugarcane. IR8 is rice. Saccharum officinarum, it's native to Brazil. Is that what you're saying, Keshav? Alright, fine. Anyways, thank you for the information. Fine. So by the use of yes, sugarcane saccharum, yes, okay. So it was not a very indigenous strain as well, but still it was very adaptable to our conditions in South India. So that is why it is said Saccharum officinarum is found in South India. I never said it's native to South India. Alright, but yes, it was native to Brazil. Yes, you're right. Alright, so when you use plant breeding to enhance the food production, it also increases the dependence on other harmful substances like fungicides and bactericides. Alright, so what is a prerequisite? So if you're going to use plant breeding to produce disease resistant plants, so what is the prerequisite there? So the prerequisite is you should know the causative organism, which organism can affect which plants, so that you should know, and how it is going to affect. For example, virus, the mode of infection is different. Bacteria, the mode of infection is different. So first you should know whether it is a virus causing the infection, the bacteria or the fungi, and how it is going to enter into your plant. So that is very, very important. Alright. For example, you should remember this. So this comes in match the following. So talking from your NEET point of view, so this is very important. So your fungi, it causes the disease called as rust. So first one is the brown rust of wheat. Then it causes red rot of sugarcane and the late blight of potato. So your fungi causes three different types of disease. One is brown rust of wheat, the next one is red rot of sugarcane and the late blight of potato. Alright, so bacteria causes only one disease, that is called as black rot of cruciferous. So remember BBC. Okay, bacteria, black rot of cruciferous. Alright, Then virus, so virus causes, you remember tobacco mosaic virus, so it is tobacco mosaic and turnip mosaic. So it is TM and TM. Fine. So now I will show you the pictures. So this is your wheat rust, okay. This is your red rot in sugarcane. So can you see red spots? So next time when you see this, remember it is your fungi that is growing inside. So please do directly eat the sugarcane when you see it like this, okay. Yes, you can definitely. If you have found me in Telegram, you can definitely ask. I will get back to you. Alright, Keshav? Fine. So this is your red rot in sugarcane and this is your late blight of potato. Then this is your black rot of cruciferous that is caused by bacteria. This is your mosaic virus. This is tobacco mosaic and this is turnip mosaic. So this is how it looked like. Fine? Okay. So the methods for breeding, method of breeding for disease resistant. So if you are going to use plant breeding for disease resistant varieties, so how are you going to do? How are you going to proceed? So the breeding or the conventional, so you can either do it by conventional or mutational breeding. Either you can take out that particular gene, disease resistant gene, and clone it inside your plant so that it gives you a disease resistant crop, okay. Otherwise you can induce mutation. So you can have two methods. So first we will look into the conventional method. So conventional method for disease resistance, you use two techniques, that is hybridization and selection. You take a plant that is resistant to disease and you take a plant that is susceptible to disease. So when you cross, the ones that are more favorable to the environment will be selected and hence your disease resistant varieties will be selected. Alright. So steps identical, the steps are very normal to your plant breeding. So we first have to do the germ plasm collection, then you have to evaluate and select your parents, then you have to cross hybridize, then choose your progeny, then you have to test and release or commercialize the progeny. So you literally follow the same steps. So there's nothing much different, okay. So these are the steps that I told you now. So first you will do screening of the germ plasm, you will understand how many type of varieties are present. Then hybridization, you first evaluate and select the parents and then you will do hybridization, then selection again you will do testing, alright. But what is the constraint here? Okay, so if you do in a conventional method, so what is the constraint, what is the challenge? The availability of limited number of disease resistance gene. Not all plants are resistant to genes, so when you're going to take a disease resistant plant, so that should be available in a large amount, right? So that itself is very limited. So that is the problem. So what is conventional technique? You have a plant A, you have a plant B. So this is very resistant to diseases and this is not very resistant to diseases. Now you're going to clone that disease resistant genes into B. Okay, so you're either going to mate A and B together to get a progeny that is very resistant in disease. But the availability of your disease resistant gene itself is less, so it cannot be, so this method cannot be applied to a wide range of plant. For example, imagine in maize, there are no disease resistant varieties naturally present. So in that case, this method, conventional hybridization method, is not possible, right? So in that case, you move on to your next method, that is mutational breeding. Okay, so now let's look into what is mutational. Okay, so before that, this tabular column is mentioned in your NCERT. So once again, tabular columns are important. So this might not be very clear. So you just have to understand the crop and what it is resistant to. Okay, so that is very important. So wheat is resistant to what now? Brassica. So brassica is your mustard. Then cauliflower, chili. So if you remember, you don't have to remember cowpea. So cowpea is not asked. So remember wheat, brassica, cauliflower, chili, and what are they resistant to. So these are the final products of your plant breeding to produce your disease resistant varieties. Alright, fine. So now entering into mutational breeding. So what is this? So you induce mutations in plants and then you screen them whether they are resistant to a particular disease or not. So for example, you induce mutation in a particular gene and you see what is the end result. So in a way, you'll know the function of your gene, right? So you can identify that function of your gene. For example, if there is a gene, okay, so this portion I am mutating. The moment I mutate this portion, the plant gets diseased. So now I will know this is the disease resistant gene. So that is how you study the disease resistant gene. Okay. So plants having these desirable characters, then they are multiplied. First you identified whether the plant has a disease resistant genes or not. Then you multiply the plant so that you get a lot of varieties. Fine. So other breeding methods among this are somoclonal variants and genetic engineering. So we'll talk about your somoclonal variations that comes under your tissue culture technique. Alright, fine. So mutation, so how do you define mutation? It is a process by which genetic variations are created through changes in the base sequence within the genes. So mutation is what? A change in your DNA base sequence that results in a phenotypic change. So that is called as mutation. Yes, Pallavi Rao, good evening. So it's good afternoon before 4. So good afternoon and thank you so much for joining my session today. So please do like, share and subscribe. That's great having you here. Fine. So how can you induce mutation? So this is mutational breeding. So you have to mutate it, right? So how are you going to induce mutation? So you can induce mutations through chemicals or radiations like gamma radiations. Ma'am, I often get confused in that table concept for 𝗃𝗃𝗃, but I don't have to remember. Yes, you don't have to remember 𝗃𝗃𝗃. So repeatedly asked one is your wheat. They ask for wheat, they ask for cauliflower, then they ask for your brassica. So remember that. You don't have to remember the middle one also. You can directly remember the first column and the third column. That is enough. Because they are not going to ask about the varieties. So you can remember this and you can remember the resistance. So what kind of disease they are resistant to. For example, we spoke about black rot. So they might ask you which variety of crop was developed which is resistant to black rot. Then you should remember it is cauliflower. Alright. So we were right here. So how do you induce mutation here? Either using chemicals or your radiations. So the process of inducing mutation and then selecting and using plants that have desirable characters as a source in breeding is called mutational breeding. So first you mutate and once you mutate you then look into that, okay. So you look into whether it is having a desirable characters and then you do the other process, so that is called as mutational breeding. So first you start with inducing mutation, then you select your recombinants and then using these plants as a source for plant breeding is called as mutational breeding, okay. No, it is not a Hindi name, it is the binomial name, okay. Might be the binomial name originated from the place where it is very common, so that is why it might sound like it is a Hindi name for you. Okay, so what are the plants that was developed by mutational breeding? So again, this example is very important. So the plants that was developed, okay, with respect, which are disease resistant, but it was developed by mutational breeding. So mung bean that is resistant to yellow mosaic virus and powdery mildew induced by mutations. Alright. Yes, Bajrang Kumar, hello. So thank you for joining my session. You say it is pure NCERT. Yes, I follow NCERT. Okay, so mutational breeding. So this is what? So initially it is very susceptible, it is highly susceptible. You start mutating a particular gene that is very susceptible. So initial and later it becomes highly resistant to a particular type of disease. So this is how your your powdery mildew looks like. So it looks like a dewdrop of powder, right? This is called as powdery mildew. Alright. Oh my god, Daniel, hello. What happened to you? Are you fine? Are you safe and sound? That's why you are late to the class. So what happened to you? Alright. So with respect to your mutational breeding, so let's look at it. So while relatives of your cultivated species of plants, they are shown to have resistant characters, but they give you low yield, okay. For example, when you do mutational breeding, what you do? You are trying to mutate a particular gene that is very susceptible to diseases or a particular pathogen, and hence you are trying to select the genes that are very resistant, right? Yes, sometimes naturally also these are present, but they give you very low yield. So naturally also you have a plant that is having disease resistant, but it's giving you low yield. Then there is no point in having a plant with disease resistant according to us, right? So the need to introduce disease resistant genes into high yielding cultivated varieties, they came into picture. Okay, now you have to combine a plant with high yield and disease resistant as well. Okay, so example, resistance to yellow mosaic virus in bindi. So bindi is your lady's finger. It transferred from a wild type. So your wild type variety had a disease resistant gene. So you transferred it into the variety that was giving you high yield. Okay, the variety that was having disease resistant was not high yielding. Thus the gene of disease resistant was transferred into a variety that was giving you high yield. Alright, one guy hit me hardly. Alright, so you were going by bike. or were you walking? So please be safe, don't fall sick, don't injure yourself, don't hurt yourself during the exam time. Alright, so it is a very crucial period where you have to prepare for your NEET exam. Okay, so you were going by cycle and who hit you? A car or a bike? By God's grace, nothing happened to me. Alright, so stay safe. Though you were going in cycle, someone hit you. Oh my God, people don't respect cyclists, right? They don't know the amount of goodness that we have when we are riding a cycle. So we are not causing any pollution, right? Yes, you were going, oh, cycle fellow hit you. Oh my God. These bike people you can never trust. The moment they have a bike, they think they have the chariot of your Ravan flying around. So what is the chariot of your Ravan called? It is called Pushpak, right? So that was a very beautiful chariot in your mythology. Alright, so please stay safe if you're riding bike, if you're going out. So please wear a helmet if you're riding bike. So that is always safe. And cycle is still safe, but still try to go towards the corner of the road. So don't say, though I am in a cycle, I will go in the middle of the road. So please don't go in the middle of the road. So try move your cycle towards the corner so that you will stay safe. Alright, fine. Yes, so the wild variety had a resistance to your yellow mosaic virus. You took that gene and you cloned it in a variety that is giving you high yield. Okay, so this is the new variety. Fine. Okay. Now, the first thing that you used plant breeding was for disease resistant varieties. The second thing that you will use plant breeding is for insect resistance. Okay, now a lot of pests are infecting your plants. So major challenge is insect and insect infestation. That is, your insect are causing a lot of infections that is bringing down your yield totally. So insect resistance in host crop plants, it can be due to morphology, biochemical or physiological characteristic. For example, sometimes if the leaves are modified to kill insects, then that is a natural modification, right? So look, hairy leaves, they are resistant to insect pests because they will poke the insect. So your insect will never come near that plant. So in a way, that plant is resistant to insects, right? So it is naturally present. So how can you make a plant resistant to insect? Whether you can modify the morphological, biochemical or physiological character. So morphology we are not going to disturb because by natural if the plant has hairy leaves, then it is resistant to insects, right? Oh my God, he came in full speed that the guy hurt his hands hardly. I don't know why people go crazy when they get a bike, but you can't help. So that is the psychology of human mind, right? So first time when people get a phone, they send you good morning, good night every day, every 1 hour. Okay, so that is the level of maturity that you have. So initially when a person gets a bike, he thinks he's on cloud 9, that he starts flying. And once or twice when he understands, no, this is not all life is about, then the maturity comes. You can't do anything, you have to tolerate such people on earth. So that is the human mentality. It's okay, but as long as you're safe, it's fine. For example, resistance to jacids in cottons and cereal leaf blade in wheat. So these are naturally occurring resistant plants. Okay. So this is your jacid. Your jacid cannot infect your cotton plant. It is naturally resistant. And this is your cereal leaf beetle. So look at the beetle. It's very unique and very, very peculiar. No, it has a lot of stuffing inside its stomach. Maybe it is eating too much. You don't know how to ride a bike, so that is also fine. It's not necessary that you have to learn how to ride a bike. Directly learn car. Fine. So in wheat, okay, the solid stems lead to non-preference by stem softfly. For example, there are a lot of varieties of insects, right? It is not like you have only one variety or two variety. You have a lot of variety of insects. For example, the plant might be resistant to some kind of insects at the same time susceptible to another kind of insects. Hi Riaz. Hello. So thank you so much for joining my session. So please do like, share and subscribe. Yes, hi. So for example, your wheat has a solid stem that makes it very, very resistant to your solid stem fly. Sorry, soft fly because of a solid stem. Then smooth leafed and nectar that makes cotton varieties very, very resistant to bollworms. Bollworms do not infect them. So high aspartic acid content, low nitrogen and sugar content in maize makes them resistant to stem borers. So these are the crops that have been developed by plant breeding which are very, very resistant to your insects. Okay. So you'll have to study the crop. You will study brassica, that is your mustard. You will study okra and you will study what kind of insects they are resistant to. Alright. So these are naturally present. For example, your cotton plant is not very, very susceptible to bollworms because they have smooth leaves and nectar. At the same time, your wheat is not very susceptible to your stem soft fly because it has a solid stem. Alright, so now I'll show you the picture. So this is your stem soft fly. He is the stem soft fly. If the stem is thick, he cannot infect that plant. So this is your bollworm. It's beautiful, no? Then this is the last one. So this is your stem boreas that lives inside your stem. Yes, so pests are always a huge problem. So finally we have gotten a solution to overcome your pest. Okay, so the last thing that we are going to look for plant breeding is improved food quality. So at the end of the day, though you cultivate, though you harvest, though you have a yield, all of us at the end of the day need a plate of meal, right? So how to improve the food quality? Even if you have one meal, people say you should have it properly. Why? Because our body is majorly dependent on a diet for a lot of vitamins, minerals, ions, etc. So the ultimate aim of all this is to one day feed everyone in the earth, to feed everyone, etc. Right? So that is why we are going to talk about plant breeding for improved food quality. Yes, after this we are going to talk about tissue culture. So diets lacking essential micronutrients, particularly iron, vitamin A, iodine, zinc, etc. Say those kind of people are very susceptible to diseases and other symptoms. So one method that we came about is biofortification. It is a breeding of crops with higher level of vitamins and minerals or higher protein and healthier fats. So what did they do? So what was the objective of your biofortification? Protein content and quality has to be increased in a plant. Then oil content and quality has to be increased. Vitamin content will be increased. Micronutrient and mineral content will be increased. Alright, so this was the main objective of biofortification, to enhance the protein and the mineral content of a particular plant. For example, if you see by biofortification, maize hybrids had twice the amount of amino acids that are present, lysine and tryptophan. So they are one of the essential amino acids that is required by your body. So that is, the amount has been increased in your maize hybrids. So if you consume this maize, then you're uptaking these amino acids, right? Then wheat variety. So the name of your wheat variety is 𝗃𝗏𝗃 66. They are having a high protein content and it is used as a donor in plant breeding techniques. Then iron, so they are trying to fortify rice as well to increase the iron content that is present. Okay, so it is fortified to have 5 times more iron content. Then crops released by IARI, that is Indian Agricultural Research Institute in New Delhi. Alright, so what did your IARI produce? They produced vitamin A enriched carrot, spinach and pumpkin, then vitamin C enriched bitter gourd, 𝗵𝗶𝗀, mustard and tomato, iron and calcium enriched spinach and 𝗵𝗶𝗀, so that people have a mix of all the vitamins entering inside their body. Hi Satyendra Kumar, you forgot the classes at 3. But anyway, thank you so much for joining my session. So please do like, share and subscribe. Then protein enriched beans, broad, lablab, French and garden peas. So I guess all the other things you know except what is batua and lablab. So I will show you. They are beans. I will show you. So this is your lablab. So they are type of your broad beans that is present. So it is very common in south. And this is your batua. They are your spinach type. They are particular type of spinach varieties. Alright. Rahul, hello. So thank you so much for joining my session. So now we are done with plant breeding techniques. So now we will enter into your single cell protein. Fortified rice has been developed to increase the iron content. Alright. Which has more fiber content, rice or wheat? Good question, you tell me. Which has more fiber content? Wheat has a lot of fiber content compared to your rice. Rice has a lot of carbohydrates. Fine. Okay, so single cell protein. So we'll quickly finish off single cell protein. Then we'll go into tissue culture. Yes, Illuminati, hello, hi. So you have been a very good audience. So without any distraction, you were listening to my class. So thank you so much. So please do like, share and subscribe the link. Alright, fine. So single cell protein. So what is the need to do single cell protein? So what is the need? We already have so many techniques. So why do you want to invest time in a lot of new techniques? That is because imagine the population is ever increasing, right? Beans are legumes. Yes, they are legumes. Alright. So the population is ever increasing and still we are not able to meet the demand of every person on earth. Hence we have to look for alternatives every day. So though you're trying to increase or enhance the production of animal meat, etc., but to feed the animal, you're again dependent on plant, right? So we tried so many techniques, but still it is a very tedious process. So how can we go about it? So understand, 3 to 10 kg of grain is required to produce 1 kg of meat. So though if you're consuming chicken, so your chicken has to be very fleshy, then only you will select your chicken, right? You will not like the chicken that is very skinny and bony. So to make the chicken very fleshy, you have to feed it properly. Again, you're dependent on grain. Again, you're dependent on the production of your plants, right? So one alternative is your single cell protein. So what is this single cell protein? So microbes grown on industrial scale can be used as a good source of protein. For example, your blue-green algae, that is spirulina, it is grown easily on materials like your wastewater from your potato processing. For example, companies which produce French fries, which make very good French fries, like your Southern Fried Chicken, your KFC, McDonald's, so they use a lot of potatoes, right? So they peel off potatoes, so the wastewater from the potato starch goes out, right? So in that water, your spirulina can grow. You do not have to invest something new from the what is existing from the waste. You can get what is beneficial. So that was the idea behind your single cell protein. It is, you are getting proteins from a single cell. So blue-green algae is a unicellular organism. Alright, it is grown easily on materials like wastewater, straw, molasses, animal manure, and even sewage. So spirulina is used in large quantities. It can be used to serve food rich in proteins, minerals, fats, carbohydrates, and vitamins. So it is available as a powder. So you just mix it with water and you drink it. Alright, so that is how you take it. So in a way, it also reduces your environmental pollution because you're not dependent on fertilizers, insecticides, fungicides, etc. So it is a very, very simple process. So this is your spirulina. Alright. Yes. This is how it is artificially available. It is commercialized as tablets also, as capsules also. So apart from spirulina, you also have bacterial species, that is 𝗵𝗿𝗏𝗏𝗧𝗏, 𝗵𝗏𝗏𝗏𝗏𝗏𝗏. Yes, someone told it, 𝗘𝗵𝗵 has told us. So this is also used to give us a high rate of biomass production and growth. So it produces 25 tons of proteins. Yes, exactly. So 25 tons from 100 grams. Alright. So it is a probiotic. Yes, good. So what is a probiotic, Illuminati? That was a perfect term, but what is a probiotic? So do you eat probiotics? When are you given a probiotic? Apart from the bacteria and the blue-green algae, you also have edible mushrooms. So since edible mushrooms are also very, very good for health, you started having large-scale mushroom industries, which is producing mushrooms on a huge scale, because that also gives you enhanced nutrition, right? Yes. Yeah, probiotic is your yogurt. So what is probiotic? Define probiotic. So these are your mushroom. So this is your bacteria, 𝗵𝗿𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏𝗏, 𝗓. Alright, fine. So finally, we are entering into what is called as tissue culture. Okay, tell me what is a probiotic. Examples are fine. Define probiotics for me. Quick. Okay, so let's go into tissue culture technique. So this is also one alternative to your plant breeding, to your single cell protein. So what else can be done is tissue culture. So tissue culture is when you're artificially working in lab with your tissues. It is called as tissue culture. Probiotics are some things from good microbes. I don't know. Okay, so probiotics. Pro is a process before, biotics is living. So in your body you have a lot of gut microbes, right? So when you're having your diarrhea, so all your microbes are lost in the watery stool. Yes. So your doctor prescribes probiotics because our body needs the growth of microorganisms to enhance the process of digestion. So when you give probiotics, it initiates the growth of other organisms. So that is called as a probiotic. So you are given probiotics so that your gut microbes are enriched and your process of digestion also enhances. So that is called as a probiotic, the one which enhances the growth of other microorganisms that are essential to the whole biosphere. Okay. 25 tons of proteins you get from 100 gram. I don't know, so it was not given in NCERT, so I will check once again and then I'll let you know. I don't want to confuse with the numbers. Alright, fine. So what is tissue culture technique? So it was one more alternative to your plant breeding technique that we came about. So what is it all about? For example, when you have a plant at home, When you go to a nursery and you get a plant, okay, so how do you start your growth of your plant? So if you have a garden, okay, so if you want aloe vera, so I have a lot of aloe vera in my garden, and if you want aloe vera in your garden, what will you do? You will cut a bit of aloe vera from my plant and you will plant it in the soil, and it obviously grows into a new plant. Very soon it will grow. So what is happening? So what is the magic behind? For example, if you cut my hand and if you put it in soil, will my hand grow into a new Krishna? Definitely not. It's possible only with plants, right? ORS is different. ORS is not a probiotic. ORS helps to balance your body fluids. Okay, so that is different. Fine. So your whole plant is regenerated from the explant. For example, if you have a rose plant like this, if you cut this portion and plant it in soil, it is able to grow into a new plant, right? So this portion is called as an explant. So any part of the plant taken out and grown in a test tube under sterile conditions in special nutrient media. So this you do it in a normal practical life, but when you do it under a test tube, when you do it under sterile condition, so that is called as a tissue culture technique. Fine. So this capacity to generate the whole plant from any particular part of your explant is called as totipotency. Totipotency is the total potency of a particular plant to develop into a whole organism. So if I take this one bit and put it into soil, you're going to have all the parts of a plant. You're going to have the complete plant together, right? So that is why it is called as totipotency. Remember, totipotency means the total potency. Okay. So nutrient media should provide, because artificially we are going to grow here, right? For example, I told you we can artificially grow your microorganisms, so we are going to use a media that is called as agar. So when I was talking about stem cell, I said we can artificially culture your cells. So again, you use a particular type of media. Similarly, when you are culturing plant cells artificially, you use a particular type of media, and that media should have a rich source of carbon, salts, vitamins, etc. for the survival of your plants. And these are plant growth regulators. So they should also be there for your enhanced plant growth. So method of producing thousands of plants through tissue culture is called as micropropagation. Okay, so a single thing can grow into a single explant. If you produce it in so many ways, it can give you a lot of plants, right? So this is called as micropropagation. Whether stem cells are also totipotent? Yes, your embryonic stem cells are totipotent. Okay, we say it as pluripotent in case of your stem cell, but your explants are totipotent. Okay. So each plant is genetically identical to the original plant, so they are called as soma clones, because you are taking a particular somatic cell and then you are growing, so it is called as soma clones. So if you are producing thousands of plants from a single parent plant, so all of them are identical, right? So you call that as soma clones, and the technique is called as micropropagation technique. Fine. So now you take this and culture, so you have a lot of cells here. So when you are working at the tissue level, you can take very, very small pieces and produce a lot of plants. So that is called as micropropagation techniques. So the plants that are produced by your micropropagation techniques are your tomato, banana, apple, etc. Fine. Yeah, so this is how it starts. First you take the seeds, okay. You put it and the seeds start germinating very soon, then it starts growing. So take it and put it in a particular test tube. So you have your soma clones here. So this is the process of your tissue culture or your micropropagation technique. So all the seeds will be identical. So all the plant that arises will be identical. Fine. So this is how you artificially grow. So another application of why you use tissue culture is because you can produce a virus-free plant. For example, if the whole plant is infected with a virus, okay. So one example of virus that we saw is your tobacco mosaic virus and your turnip mosaic virus, right? So one unique feature of your virus is your virus cannot infest or infect on your meristematic cells, okay. But our book tissue culture is one chapter. Alright, so you might be following state board chapters, right? Alright, fine. So that is also good, but for NEET level you don't have to go into much depth, so that is also better. But the more you know about a particular topic, the best it is, so you will know in and out. So one thing that is, your virus cannot survive in a meristematic division because your meristematic zones are the ones that actively divide. Your virus cannot actively undergo division, it's a bit slow, okay. So it cannot survive in a meristematic region, so thus your meristematic tips of your plants are very free from virus. For example, your apical and axillary meristem, they are very free from virus. Even if the whole plant is infected with virus, if you take the meristematic region and do a tissue culture process, you will get the new plant that is free from virus. Alright. So meristems of banana, sugarcane, potato were successfully tried. So you take the meristematic region. So this is the meristematic region. and it produces a new plant that is free from virus. Alright, so that is about your meristematic culture. Coming to what is protoplasmic fusion. So what is protoplast? You have your cell. So the outer layer is your cell wall. So if I remove the cell wall of your cell, the other intact cell material is called as a protoplast. So if I take two different cells and if I destroy the cell wall, whatever I will be left with is your protoplast. If I fuse these two protoplasts, I am getting a new plant, right? So it is a hybrid plant. So protoplast, when I fuse the protoplast of two different cells, I get what is called as somatic hybrids, okay? So I am not doing anything with the gamete cells. I am not doing anything with the germ cells. Instead, I am taking the entire somatic area. Instead, I am taking the entire protoplastic area. So I am taking the protoplast from two cells and I am trying to hybridize it. So it is called as somatic hybrids. So end product is called as somatic hybrids and the process is called as somatic hybridization. One example is your tomato. Yes, you're right, Illuminati. So tomato into potato gives you tomato. So how that happens? So see, so you have isolated cells. So say this is potato and say this is tomato. Fine, you take the cell wall out, so the intact cell wall material is your protoplast. So you take the protoplast and then you culture it artificially on the cell. Then you try to hybridize both the protoplasts together. You induce fusion of protoplast. You try to join the two protoplasts, so you have agents that will join two protoplasts together, alright, like your pig. So once it joins, so you have two protoplasts coming together. So it will have all the characteristics of both the plants, because this plant will have particular type of genes, this plant will have particular type of genes, this will have a particular concentration, particular secretion, so this will have another one. So when you try to fuse everything in the protoplast, it will start producing hybrids. So these hybrids are called as somatic hybrids and the process is called as somatic hybridization. Alright, was tomato successful? Not to a very good extent. Yes, you were able to fuse protoplast, so that was understood. But your tomato was not successful, it was not healthy either. Alright, why root is not decomposed in soil? It depends. Sometimes, yes, it decomposes in soil. Who told you it does not? For example, but your roots are very, very rich in minerals, right? Your root also has your meristematic region. So you have your cells present. So quickly your root also grows. It does not decompose very quickly. Your root does not give up very quickly because it has your meristematic cells which undergo division and which tries to give life to the plant. So that is why it is not easily decomposed. But yes, it decomposes, okay? When it is a very old tree, yes, it decomposes gradually. Alright, so with this we are done with your strategies for enhanced food production. Alright, so if you genuinely like the session, so please do like, share and subscribe. So we are done with this unit also. So in this unit you had human health and diseases and you had microbes and human welfare. Then we spoke about strategies for enhanced food production. So in Monday's class we will discuss the questions from these three units. But before that, tomorrow we have a class 11 session that is going on at 3 p.m. So we will discuss more questions from your biological classification and tricks to remember the examples in biological classification. Alright, yes. So if there is a mentee tomorrow, if there is a special lecture tomorrow, I will let you know. But until then, as of now, you have a lecture tomorrow at 3 p.m. So please join. Before we start our next chapter, I will finish the questions from this unit and then we'll go ahead with the next chapter. So next chapter will be from ecology. Alright, yes, because someone was asking about ecology. So we'll go ahead with ecology. So ecology is way, way interesting. So we'll talk about ecology. Alright. So thank you so much for joining my session today. So have a great evening ahead and this is me signing off. So take care and bye-bye.